Okay, everybody, welcome to part C of our chapter dealing with the central nervous system, where we're going to get into more discussion of some of the functional brain systems. Okay, these are networks of neurons that that connect large areas of the brain and share certain sets of function. Examples include the limbic system and the reticular formation. Now, the limbic system you could best describe as our emotional brain. And so, what it does is it ties emotional content to our experiences, and it also lets us express emotion. If an individual has a limbic system that's poorly developed or non functional, then they would have a difficult time um, assessing emotions in the facial expressions and the actions of others or expressing emotions themselves. An example where this takes place is in a condition known as autism. The reticular formation's job is, among other things, to control sleep and wakefulness through what's called the reticular activating system. Okay? The reticular activating system utilizes the thalamus, which is the gateway to the cerebral cortex, and together with production of melatonin, is designed to control how aware we are of everyday stimuli going on around us. Okay, melatonin levels um, are going to drop when the sun is up, and they're going to rise when the sun is down. The idea is that the eyes perceive the light levels. That is connected via the hypothalamus to the pineal gland, and that controls melatonin production. And the melatonin levels, along with the reticular activating system in the thalamus, determine to a large extent what stimuli make it to the cortex and which don't. The limbic system is a set of structures on the medial aspects of the cerebral hemispheres and diencephalon, and there are also some structures that encircle the brainstem. Okay, there are components of the diencephalon, there are components of the cranial nerves, um, and structures such as the hippocampus and the amygdala that are part of this system. Okay, so you can see them here. They surround the thalamus, and they surround the lateral ventricles. Okay, and you can see the structures here in bold. Okay, um, all with different roles. Okay, and we'll address those roles as we move through this system. Okay? The limbic system we can think of as our emotional brain. The amygdaloid body is going to recognize anger or fearful facial expressions. It can assess danger and it can elicit the fear response. And it does that utilizing things like the hypothalamus. Okay. The cingulate gyrus allows us to express emotions through gestures and can resolve mental conflict. It also is responsible for putting emotional and memory content to odors. Okay. The olfactory bulb and tract and the olfactory nerve are part of the limbic system, and they are strong elicitors of memory, okay, and of course the emotions that come with those odors as well, okay. An example: skunks smell bad, um, roses smell wonderful. The idea of bad or wonderful is a result of an assessment of a limbic system input. Okay, if you didn't have a functioning limbic system, you would simply say that a skunk smells like a skunk and a flower smells like a flower. Okay, most of this output goes through the hypothalamus, a region of the brain under the thalamus and above the pituitary gland. The limbic system also interacts with the prefrontal lobes so that we can react emotionally to things that we consciously understand are happening to us, and it allows us an awareness of the emotional content in our lives. The hippocampus is like the body's DVR, and what it does is it records your experiences during the day, and then when you dream, um, that's part of the role of the hippocampus, storing that information into association areas in the brain as memory. Okay, the amygdaloid body has a role in this as well. Individuals with a deteriorating hippocampus often exhibit signs of senile dementia. Okay, the reticular formation are broad columns of gray matter that run the length of the brainstem and include the Rafe nuclei, the medial nuclei, and the lateral nuclei. The reticular formation has connections to the hypothalamus, the thalamus, and cerebral cortex, as well as the cerebellum and spinal cord, and one of its big roles is to control sleep and wakefulness. Okay? The reticular activating system is designed to control what we're consciously aware of. Okay? What it does is it controls whether or not the thalamus can direct stimuli that come up through the spinal cord and brainstem to the proper cortex in the cerebrum so that we can assign meaning to it using our association areas and become aware of it. Okay? If the thalamus doesn't let that information up to the cerebral cortex, then we're unaware of it. Okay? And the reticular activating system is responsible for controlling the stuff that we're consciously aware of, as well as everyday stimuli that are weak or repetitive, which incidentally is 99% of the stuff that goes on around us. So it's kind of a noise filter. It's inhibited by sleep centers, alcohol and drugs, and severe injury to it can result in coma. So the idea here is that the activating system is part of the gate opening device for the thalamus. Okay? Motor functions, it helps control coarse limb movements through the reticulospinal tract, and reticular autonomic centers are going to regulate visceral motor functions. Examples include the vasomotor centers that control vasomotion and blood pressure, and the cardiac center that controls the rate and force of heart contractions. The respiratory centers are designed to control the rhythm of breathing, and their most important role is to control breathing when we're unconscious. Okay? The pontine centers in the pons and the ventral and dorsal respiratory groups in the medulla are part of these nuclei, all right, that help to regulate our breathing pattern. The brain is protected by several layers. It is a vital organ. So the brain, brainstem, and spinal cord are all surrounded by bone. They all float in cerebrospinal fluid, which is a filtrate of blood, and they are all cloaked by membranes called meninges, which from the outermost to the innermost aspect include the dura mater, the arachnoid, and the pia mater. Okay? And these meninges also stabilize the position of the central nervous system structures that are floating in the cerebrospinal fluid so that they don't bump and damage themselves against their hard outer coverings. Okay? So those are the meninges and cerebrospinal fluid. The blood-brain barrier is created by the ependymal cells and the astrocytes, and we sort of touched on it when we were talking about nervous tissue earlier. And the idea is to isolate the central nervous system from harmful polar material that would be in the blood supply. So what this does is this locks out a lot of toxins and pathogens, and as a result keeps the brain relatively safe. The brain and the whole central nervous system have very little immune function. Okay, the microglia are basically it. So anything that penetrates the blood-brain barrier is going to do a lot of damage. Okay, and there are vulnerable spots that some pathogens are able to penetrate. We should also point out that non-polar material goes right through the blood-brain barrier and into the central nervous system. Examples include anesthetics and alcohol. The meninges cover and protect the central nervous system, as well as protecting blood vessels and enclosing venous sinuses. They contain cerebrospinal fluid, which is a filtrate of blood. CSF is manufactured in the choroid plexuses of each of the ventricles in the brain. The meninges also form partitions in the skull. The three layers are the dura, the arachnoid, and the pia mater, and that's from superficial to deep. Okay, meningitis is simply an inflammation of the meninges, and it's an extremely dangerous condition. It can be brought about by trauma, toxins, bacterial or viral infection. And the danger, of course, is that if there is a pathogen or a toxin involved, there is relatively little immune defense against its effects. The dura mater is the most superficial and the strongest membrane. It's a double layer of fibrous connective tissue around the brain that separates in the dural venous sinuses, which is a space that's between the cerebral hemispheres. The dural septa limit too much movement of the brain, and they anchor the brain to the floor of the cranium, okay, so that it's not just bobbing around aimlessly inside your skull, okay, it's tethered. The falx cerebri is in the longitudinal fissure and is attached to the crista galli, which is that rigid bone that is right in the middle of the top of the ethmoid and surrounded by the cribriform plate. Okay? The falx cerebelli is along the vermis of the cerebellum, and the tentorium cerebelli is a horizontal dural fold over the cerebellum and in the transverse fissure. Okay? The arachnoid mater is the middle layer, and it gets its name from the web-like extensions that run throughout it. It's separated from the dura by the subdural space, and the subdural space is filled with cerebrospinal fluid and invested with large numbers of blood vessels. Okay? The arachnoid villi protrude into the superior sagittal sinus, and that allows the cerebrospinal fluid to be reabsorbed after it has circulated through the ventricles and around the entire central nervous system to go back into the blood supply and become part of the venous return to the heart. The pia mater is the deepest meninx, and it clings very tightly to the brain, and it has lots of blood vessels in it. Okay, it's extremely delicate. All right, but it is a critical meninx because it helps to anchor the extensive blood supply close to the neural tissue that requires it. Okay, so you can see the protective layers here, okay, from superficial to deep. Notice the arachnoid granulation, the villi, right? There's the superior sagittal sinus, and then those are the web-like extensions of the arachnoid, and this is the subarachnoid space here with the CSF in it, and then that's the pia clinging to every convolution. And again, under that gray matter and deep to that white matter, right? This is a coronal or frontal section through the skull, right? So that you can see where all these structures are located. Cerebrospinal fluid is manufactured from blood. It is a filtrate of blood made by the choroid plexuses in the roofs of each of the ventricles of the brain. It has less protein and a slightly different ion concentration than plasma. It's a constant volume because as it's created, it also drains back into the blood supply in those arachnoid granulations, right? It goes back into the dural venous sinus and then becomes part of the blood supply that goes back to the heart. What does it do? Well, it allows the central nervous system structures to float, okay, reducing their weight by 97%. It protects the central nervous system from trauma, and it nourishes the brain and carries chemical signals, all right? And it's a liquid, so because it has a lot of water in it, part of what it's able to do is indicate to structures, for instance, in the brainstem and the hypothalamus, the chemical changes that occur in the blood supply as a result of nonpolar material being able to diffuse across the blood-brain barrier, such as CO2 and oxygen. Okay, you can see how the cerebrospinal fluid is manufactured. Okay, how it flows from the lateral ventricles through the interventricular foramina to the third ventricle, which you can see here, down the cerebral aqueduct to the fourth ventricle, and then continuing down the central canal of the spinal cord, which goes right through the middle of the gray matter, but also out of median and lateral apertures to bathe the entire outside of the central nervous system and then be reabsorbed in the dural venous sinus at the arachnoid granulations. We noted earlier that the choroid plexuses are found on the roof of each of the four ventricles, and they generate the cerebrospinal fluid by filtering blood. These capillaries are enclosed by pia and ependymal cells, which use ion pumps, and that controls the composition of the cerebrospinal fluid, and it cleanses the cerebrospinal fluid by removing waste products. So the idea here, again, is to create an environment in which the neurons can work most effectively. So the cerebrospinal fluid maintains a temperature, a pH, and an ion concentration that's optimal for the neurons to do their job. Normal volume is about 150 milliliters, and that's replaced every 8 hours. And you can see here a diagram of the area of production of cerebrospinal fluid. This might be a section of choroid plexus in any of the four ventricles. And you can see the ependymal cells and the tight junctions between them, right? Tight junctions and desmosomes. And so the only way that the filtrate gets into the cerebrospinal fluid is through these cells themselves, okay? So the ependymal cells and astrocytes are in large part responsible for the composition of the cerebrospinal fluid, okay? Occasionally what will happen is that the circulation of cerebrospinal fluid can be blocked. In newborns, this results in an expansion of the cranial vault because of the increased pressure due to the buildup of cerebrospinal fluid, and since the cranial bones have yet to fuse at this point, the vault will enlarge in order to compensate for the increased pressure. The fix for this is to put a shunt that runs from upstream of the block of the cerebrospinal fluid down into the abdominal pelvic cavity, and the excess fluid can then be absorbed by the capillaries that are in the peritoneum. That relieves the pressure in the cranial vault, and then the skull can go back to its normal dimensions. And then this has to be kept in the entire life of the individual in the event that there's a block subsequent to that. Now, if this were to occur in a normal individual after the bones of the cranium have fused, this could result in coma and death, okay, because there would be nowhere to relieve that pressure, and you would simply crush the neurons of the brain. So it would result in a condition not unlike what happens when people have a concussion. Okay? The blood-brain barrier we've discussed, right? And this is simply a way to protect the brain from polar material that represents a possible hazard. Okay? Toxins, pathogens, and so on. Okay? It maintains a stable environment. It separates the neurons from those blood-borne hazards we discussed. And what is it made of? It's made of the astrocytes and the ependymal cells, right? The endothelium of the capillary walls is shored up by the fact that the basal lamina around the capillaries that supply the brain is thick, but at the feet of it, the astrocytes generate signals so that tight junctions are formed in the capillaries that invest their supply into the central nervous system structures. Okay, so what you have here is a connection between the blood supply and the neuron, mediated by the astrocyte, but the formed elements of blood and polar compounds do not come into contact with the nerve tissue unless the astrocyte deems that it's okay. Okay. And the same is true of the ependymal cells, right? The ependymal cells are not going to let anything into the cerebrospinal fluid that could represent a hazard, all right? It's a selective barrier. It lets nutrients move through by facilitated diffusion. Metabolic waste, proteins, toxins, drugs, and non-essential amino acids, as well as potassium, do not pass through, okay? It allows any fat-soluble substance to pass, including alcohol, nicotine, and anesthetics, right? It's absent in some areas, such as the vomiting center and in the hypothalamus, and the reason being that we have to monitor the chemical composition of the blood here, but these also represent vulnerable regions where pathogens and toxins can invade, okay? Traumatic brain injuries are simply physical insults to the brain. Examples include concussions, where we have temporary change in function, a contusion, which is permanent damage. Subdural or subarachnoid hemorrhage is bleeding, in the case of subdural under the dura mater, and in subarachnoid, it's going to be in the subarachnoid space. Okay, it can force the brainstem through the foramen magnum, can damage those structures, and since that is a brainstem structure, can result in death. Cerebral edema is a swelling of the brain that's often associated with traumatic injury, while cerebrovascular accidents include ischemia, which is temporary loss of blood supply to brain tissue that can die. Okay, this can be the result of an embolism or the result of a blockage. Okay, and then a TIA, where we have a temporary loss of oxygen to a region of the brain, and that produces a reversible ischemia. Okay, but a TIA is often a precursor to a stroke. Degenerative brain disorders include Alzheimer's disease, where we see death of neurons, okay, especially in areas that are important in processing memory, okay. What happens as neurons die is it becomes more difficult to access information stored in the brain, and in many cases, some of that information gets destroyed, and the result is that the person becomes less and less familiar with his or her surroundings, and they start to lose skill sets. Okay, we see disorientation, language loss, moodiness, confusion, hallucinations. One of the hallmarks of Alzheimer's are beta amyloid plaques that form in the brain. We don't know whether this is a cause of Alzheimer's or an effect of Alzheimer's. We just know that it is a hallmark of the disease. This may be um, a disease that operates similar to the way that prions operate. Okay, prions are small proteins that can cross the blood-brain barrier because of their tiny size, and they can misfold proteins that are called chaperones, which help other proteins fold up properly, and the result is degenerative disease that causes the neurons to die. Okay, we don't know, but this is speculative. Neurofibrillary tangles inside the neurons inhibit their function, and again, we don't know whether this is a cause or an effect of the disease. The brain will shrink. Okay, in Parkinson's, we have death of the substantia nigra of the midbrain, and we lose the dopaminergic neurons there. The basal nuclei get deprived of the dopamine, they become overactive, and you have resting tremor. You have ataxia, okay, and we have difficulty starting and stopping voluntary movement. All right. Treatment, oral L-DOPA and deep brain stimulation. We unfortunately don't have a cure. Okay. The L-DOPA is sort of a double-edged sword because the brain can, in many cases, adjust to the L-DOPA orally and become less sensitive to it, and then the effects of treatment with L-DOPA become less and less effective. There's an excellent movie about this disease called Awakenings, starring Robin Williams, that's worth a look, and it talks about the discovery of the use of oral L-DOPA for the treatment of Parkinson's, and how initially the results were quite promising, and then the sad result of many of the patients is that they went back to their previous state. Okay, so take a look at that if you get a chance. Huntington's disease is a fatal hereditary disorder caused by a protein called huntingtin, which is made improperly. Okay, this leads to degeneration of basal nuclei and cerebral cortex neural tissue. We see initially jerky or flapping movements, but eventually we end up with mental deterioration and early death. Okay, so what happens here is you do lose cognitive function, And at this time, the end the end game is always early death. Okay, the tragedy of this disease is that it is a hereditary disorder that is inherited as an autosomal dominant and does not show up until late adulthood. So if you have a parent with Huntington's, the children of that parent have a 50% chance of having Huntington's. And we can easily detect whether or not they have it. And many of the people that discover this often decide not to be tested for the disease because they don't want that to dictate the way they lead their lives. We can ameliorate some of the symptoms with drugs that block dopamine effects, and we are investigating the effect of stem cells on this disease. Some pathways that are worth knowing in the brain include the motor pathway that talks to that goes from the cerebral cortex, the upper motor neurons, to the cerebellum, and the lower motor neurons to the muscles. We call those the pyramidal tract. And then the control of movement via the cerebellum that governs a lot of these motor action plans that get sent down. And then we're going to talk about some sensory pathways as well. So the first pathway we're looking at here is the generation of a motor action plan. Okay, so what happens in the premotor area in the frontal lobe is that we decide based on stimuli that we've received from other regions of the cortex what we want to do. Okay, that activity gets moved to the primary motor cortex, which is in the precentral gyrus, and then what has to happen at that point is that we have to relieve the blocking effect of the globus pallidus. All right, so the way that works is that the other basal nuclei, along with the midbrain, act to remove the inhibition of the globus pallidus, and that stimulates the neurons in the precentral gyrus, and that allows them to fire. That information then goes down the pyramidal tracts pictured here, and then we hit lower motor neurons, and then they head out to the skeletal muscle, and we do our thing. Okay, so the idea here is that we have to have cooperation between the midbrain and the basal ganglia and the thalamus in order for the cortex to do its thing. Okay, so ultimately, once we remove the blocking effect of the globus pallidus, the thalamus can give the go-ahead for the cortex to fire, and then those instructions can head down the pyramidal tracts to the brainstem and the spinal cord. Okay, remember I told you that the thalamus is the gateway to the cerebral cortex, so it is responsible not only for directing the stimuli to the proper cortical region for sensory stuff, but it also opens or shuts the gateway to letting the motor pathway fire. Okay, so it's sort of like getting an okay to go ahead and do the action plan. Now, when that happens, some of that information in the precentral gyrus is going to go to the cerebellum. Okay, the cerebellum will then compare that information to sensory information, and if there is an agreement, everything's cool. If there's a disagreement, what will happen is that the cerebellum, because it's using input from your balance, your proprioception, and your special senses, okay, will then adjust that motor action plan, send that information back to the thalamus, and that'll send that information back to the cortex on the precentral gyrus, and then we will repeat the process of sending that information down to the brainstem and the spinal cord. Okay, so the cerebellum here acts like an editor of our movements in order to both coordinate our movements and make them fluid, but more importantly, to make sure that our motor intentions match our motor actions. Okay, so that is its critical role. Another example of a pathway, sensory pathway, is visual perception, and then the movement of the eyes tracking the object that we're looking at. So the first pathway is relatively easy, right? The retina is going to perceive light bouncing off an object and send that information via the optic nerve, which is cranial nerve 2, down the optic tract to the thalamus, okay? The lateral geniculate nucleus of the thalamus contains neurons that will then project to the occipital cortex at the tip of the occipital lobe, and that will be sensation that we're seeing something, and then the perception of what we're seeing is drawn from the visual association area in the occipital lobe. And then that information is also going to be routed to the superior colliculus, which is on the midbrain, okay? And the retina, along with the occipital cortex, okay, will have output to the nerves that move the eyeball, all right? And so that's the oculomotor, abducens, and trochlear nerves. That will allow us to track the object that we're looking at, okay? The frontal eye fields also play a role in this, okay? Now, this is something that you do almost automatically. You don't even really think about it that much. Um, if you want, look at an object across the room right now, okay? And then, fix on it and then move your head, okay? And as you move your head, what you notice is that the object in your visual field isn't moving, right? But your head's moving. So why isn't the object moving? Well, the object isn't moving because your eyes are compensating for the movement of your head. So this is how you're able to run or move and keep a steady image in your mind of whatever it is you're looking at. Otherwise, if your eyes didn't do that, everything would be like a swirling camera. Everything would be a blur, okay? Kind of like the Blair Witch Project, okay? So the idea here is that these are automatic adjustments in our gaze that take place in order to allow us to fix on an object at a distance. So this is one of the things that helped us become hunter-gatherers, right? We were able to look at an object at a distance. The fact that our eyes are in the front of our head provided us depth perception, and so we were able to see how far away something was that we wanted to kill and eat, or how far away something was that represented a hazard, okay? And we could run from it. Another example of this that you're unaware of is the fact that even when you think you're looking at an object that is fixed in your visual field, the fact of the matter is that there are tiny movements of the eyes that take place when you look at that object that you're kind of unaware of, but these pathways govern it, all right? And it prevents the bleaching of the photoreceptors so that you lose your visual field. So if you meditate really hard and lock your gaze on an object consciously, okay, what you can do by locking your gaze is you can watch your visual field vanish and you will temporarily go blind, okay? And of course you'll recover your vision when you move your eyes around. So if you want to try that, you're welcome to do it, okay? All right, that brings us to the end of Part C. I will see everybody in Part D.